If you don't understand VLDL, you'll never understand LDL. VLDL metabolism represents the endogenous pathway of triglyceride transport, where the liver exports internally synthesized lipids to peripheral tissues. Step 1 begins in the liver. Hepatocytes assemble and secrete nascent VLDL particles, which are rich in triglycerides and contain Apo B-100 as their structural apolipoprotein. At this stage, VLDL is immature and not yet fully functional. In step 2, once VLDL enters circulation, it interacts with HDL. From HDL, it acquires Apo C-II and Apo E, converting it into mature VLDL. Apo C-II is crucial because it serves as a cofactor for lipoprotein lipase, while Apo E will later mediate remnant uptake. Step 3 occurs at the capillary endothelium of peripheral tissues, especially adipose tissue and muscle. Here, lipoprotein lipase, LPL, anchored to the endothelium, is activated by Apo C-II. LPL hydrolyzes the triglycerides within VLDL into free fatty acids and glycerol. The free fatty acids are taken up by tissues for storage or energy, while glycerol is transported back to the liver. As triglycerides are progressively removed, VLDL becomes smaller and denser, forming intermediate density lipoprotein or IDL. In step 4, IDL has two possible fates. A portion of IDL returns Apo C-II and Apo E back to HDL and forms LDL with only Apo B-100. About half of IDL is taken up directly by the liver via Apo E-mediated receptor binding and undergoes further triglyceride depletion primarily through hepatic lipase activity. Finally, in step 5, LDL delivers cholesterol to peripheral tissues. It binds to LDL receptors via Apo B-100, leading to receptor-mediated endocytosis. LDL can also be taken up by the liver for recycling or excretion. Clinically, this pathway is critical because elevated LDL levels are strongly associated with atherosclerosis, while defects in LPL or Apo C-II lead to severe hypertriglyceridemia. Thanks for listening. Like, share and follow the channel to keep learning biochemistry the clinical way.